Hello students, welcome to Trunkie Academy. We are studying about in the last class and the previous class we studied about the first we started with the diversity in living world, and then started with some other topics, that is the group of different animals, plants, okay, and so on. And now today we'll study with and after that we'll also study about the cell, the structure and basic unit of life. And after in that particular chapter we study about the prokaryotic and eukaryotic cells and the different important cell organelles. Okay, now today another very important chapter, and the different topics are there in this chapter, that is the biomolecules. Biomolecules is one of the important chapter in which we study about the different important biomolecules, that is carbohydrate, lipids, protein, fats, etc. Okay, so uh, the most important in fact all the important uh, biomolecules play some important role in physiology of the body. Okay, so all the molecule that is present, okay, the macromolecules and the chemical compound that is present in our body plays an important role. So we'll start with the amino acid, and here first we'll start what is biological compounds. Biological compound, and there are different types of amino acids are organic compounds containing an amino group and an acidic group as substituents on the same carbon. Okay, so that is the alpha carbon. What are amino acids? So amino acid, that means the protein, and that plays a very important role in the building block, or you can say the contributing the mass, or contributing the muscle mass of the animals, or even in plants also we find proteins are present. Okay, so what happens, amino acids are organic compounds containing an alpha amino group. Okay, so in protein what happens, there is amino acids as a monomeric unit and connected by another monomer or monomeric unit of the amino acids, and so on. It will be connected with the peptide bonds, connected with the peptide bond. Okay, so this forms a polypeptide chain, and the one monomeric unit, okay, one monomer of these proteins is the amino acid. So proteins are made of the monomeric unit of amino acids, and these are organic compounds. Organic compounds mean where we find the carbon, hydrogen, okay, and there's so many different types of organic compounds are there, and these organic compounds are found in the living organisms. Okay, so and then some as the substituents and the same carbon, and okay, so amino acids are organic compounds containing an amino group and an acidic group. So there will be one amino group and amino group and acidic group. Okay, here you can see amino group and the acidic group. So this is the acidic group. Okay, carboxylic acidic group, and here is the amino group. Okay, so now you see this that and group as substituent on the same alpha carbon. So they both are attached to the same alpha carbon atom. So this is the alpha carbon atom. See here, this one. Okay, so this is the alpha carbon atom, and both are attached to the same. The amino group here, this one, the yellow shown in the here, this one, this group and this, both are attached to the same alpha carbon atom. So amino acid are organic compounds containing an amino group that has been shown in the yellow, and one that is with the acidic group that is shown in the red, and both are attached with the same alpha carbon atom, and there is one kind alkyl group also. Okay, and hydrogen. Now this is the structure of the one a typical, or you can say the diagrammatic, you can say amino general structure of the amino acid. Okay, so alpha carbon. Okay, alpha carbon. Hence, amino acids are called alpha amino acids also, okay, because it is attached with the alpha carbon atom, so it's also called alpha amino. Amino acids are substituted methanes actually, okay, substituted methanes, and methanes CH4, okay. So here you can see methane CH4, okay. So methanes are actually with the CH4, and therefore you can see that this is the structure of the methane, and here you can see that there are 4 substituent groups occupying the 4 valency position in case of the amino acid. So these are hydrogen is there, carboxyl group R, okay, in case of the R is there, and amino group is there, and a variable designed R group is there. Okay, so you can see the carboxyl group means the acidic group, the acidic group. Amino group R is there, R group, and variable R group is there, amino group is there. So in the previous slide we saw that here is the variable R group is there, here was the amino group was there, amino group, and here it was the acidic group. Okay, and this was the hydrogen. So you can see this was the actually amino acid is replaced, or you can say substituted methane. Okay, where we find there is the substitution of the H hydrogen atom by 3 different group, carboxyl group, and then amino group, and a variable designated R group. Okay, now designated R group is a variable group. So that is the, you can see in the diagram the R, the side chain variable, which is designated as R. Okay, denoted by R. Okay, alpha carbon, then amino group, and then the carboxyl group, acidic group. Okay, so this side is the carboxyl group or acidic group. Okay, this is the side, and here we find the amino group. Okay, and this is the variable group designated or denoted by R. Okay, so based on the nature of R group, there are many amino acids. So the what brings the changes in the property and the behavior of the amino acid that is decided by the R group. There are many amino acids. Okay, so what happens? Amino acids which occur in proteins are only 21 types. So try to understand this. So we have got same things here in the structure. We'll find, there are R group and the amino group and the carboxyl or acidic group, but the R is varying, and then that is changing the property of the amino acids, and based on this there are different types of amino acids. Amino acids which occur in protein are only 21 types. So in case of proteins, okay, it is only 21 types, and these 21 types, they with the help of with the permutation and combination, they form the different types of protein. Okay, with the help of permutation and combination of these 21 types, there are different types of proteins are formed. The R group in these proteinaceous amino acids could be a hydrogen also. So like the I. So if that R is replaced by the H only, okay, not a CH4 or CH3 or CH2, it's only H, then it is the simplest amino acid, and that is the glycine. Okay. Now a methyl group, it is then alanine. If it is the hydroxyl methyl, then it's serine, and then here you can see that this is the glycine. This is the glycine, right? Because it is the H only, hydrogen atom. Okay, so no bulky glycine. Now in case of CH3, then it becomes the alanine. If it is the hydroxyl methyl, then it is the serine. Okay, so this is how there is different kinds of amino acids depending on the one group, the R group, which is changing, the variable, and that is actually deciding the property of amino acid. Okay, the chemical and physical properties of amino acid are due to amino group, a carboxyl group, and R functional group. So these 3 things are actually deciding, or you can say the chemical and the physical properties of amino acids are due to the presence of this amino group, carboxyl acidic group, and this R functional group. Okay, the functional group. Okay, the changing variable one. Based on the number of amino and carboxyl groups, so try to understand. Based on the number of amino and carboxyl group or acidic group attached to the amino acids are acidic amino acid. Okay, yeah, here you can see the aspartic acid. Okay, aspartic. So here you can see the 2 carboxyl group, right, and 1 amino group. Okay, basic amino acids where we find the base. Okay, histidine, lysine, arginine. These are the basic amino. Neutral amino acid. You can see here. Glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine. These are the your neutral amino acids. Okay, so these are about five, nine neutral amino acids. So aromatic amino acids are the amino acids that include aromatic ring, simple benzene ring. Okay, so tyrosine, phenylalanine, tryptophan. These are the three aromatic group. Okay, so I mean with the because of the presence of the benzene ring, or you can say aromatic ring. Okay, so that is the aromatic amino acids. Now, what is deuterion? This is a very important for exam, for the NEET exam. Deuterion, very important for exam. Okay, important. Deuterion. A molecule such as an amino acid that carries both positive and negative charge is called deuterion. Okay, so we find that in case of amino acids, okay, we see that there is in the same molecule of the amino acid, there is one part, one region has negative charge and one has got the positive charge. So one molecule like amino acids that carries both positive and negative charge in a single molecule, that is a deuterion. Okay, like amino acid structure, you can see that here is the N plus in the amino group, and you can see the O minus in the solid state amino acid exists in dipolar or deuterion form. A deuterion is a neutral molecule with a positive and a negative electrical charge. Okay, so it is a neutral molecule with positive and negative electrical charge. Okay, so this is the neutral molecules. Okay, it's called deuterion. The particular property of amino acid is due to the ionizable nature of amino and carboxylic groups. The particular property. Okay, because we say there are different kind of property is exhibited by different amino acids, and this is due to the ionizable nature of amino and carboxylic group, because they are easily ionizable. Okay, ion furnishing. Okay, amino acid can have several forms depending on the pH of the system. Yes, amino acid when it is in the neutral pH, then it will behave, or it will have little different kind of behavior, and when it will in the acidic medium, then it will have little different behavior, and when it is in the new alkaline pH, then at low pH or acidic condition, amino group is protonated by addition of proton from the acids. You know, the acids are having H plus ion, okay, proton ion, so that is getting added. Okay, so it is protonated. Amino group is protonated, and at low pH it is the now at high pH or basic conditions, okay, the carboxylic acids, okay, is deprotonated by the removal of proton. So what happens in case of the alkaline pH, okay, or basic conditions, the carboxylic acids is deprotonated. So one proton is removed, removal of proton. Okay, so that way they behave. So you can see the cation, okay, the low pH. Then in the deuterion as the normal pH, at the neutral pH. Okay, and in case and the anion is formed in case of the high pH. So one amino acid can behave like a cation in the low pH, in acidic medium, or in acid. Okay, due to protonation, because the H plus ions are then protons are there in the acid. And in case of the high pH or alkaline pH, we can see the there is a removal of H plus. Okay, here there is the removal of the H plus. Okay, and this causes the formation of anion. Okay, so that is how the formation of cation, and hence in solution of different pH, the structural amino acid changes. They behave differently, and deuterion when H plus in aqueous solution, okay, with the help of this protonation, it now a positive ion overall charge of +1. It's no longer a deuterion. So deuterion is the amino acids is the deuterion only at the neutral pH. Okay, so when it is protonated, it becomes no longer a deuterion. It will behave as a cation. Okay, so at low pH or acid conditions, and here in case of the high pH or alkaline pH or basic conditions, you can see it behaves as a anion and no longer a deuterion, because it has overall -1 charge. Okay, now we'll go through some questions. Amino acids are organic compounds containing hydroxyl group. amino and acetic group, phosphate group, or sulfate group. It is the amino and acetic group, or you can say amino or carboxylic group. So number 2 is correct answer. Amino acids which occur in the proteins are only of 19 types, 18 types, 10 types, 21 types in the protein. Okay, so many different types of proteins are. Keratin is there. The proteins that is present in the muscles are there. Okay, present in the nails are there. Okay, so many different proteins are found in the living organism. These all proteins are made of 21 types of amino acids. Okay. Aromatic amino acids are glycine, alanine, valine, or phenylalanine. It is a phenylalanine which has got the aromatic ring or benzene ring. Okay. Now biological compounds, lipids, and heterocycline. Now we study about the amino acids, and the later on we will also study about the protein. Amino acids means only the monomeric unit of protein, not protein. Okay. When these monomeric units, amino acids are joined by peptide bonds, they form a long polypeptide chain, and these chains are again joined side by side with some kind of bond like sulfide bonds. Then they form a large macromolecules and becomes protein. So protein has got a different structure, and while forming the large macromolecules, these amino acids together in the form of protein, they act as certain compound, chemical compound, and they form the basis for the important functions where they are present. Okay, in the in living body. Now lipids. What are lipids? Lipids are class of organic compounds that are fatty acids or their derivatives. Okay, so they are the fatty acids or their derivatives. They are insoluble in water but soluble in organic solvent. Try to understand this. These fatty acids, oil and fats, they are not soluble and not easily miscible with the water, but that can be dissolved. Okay, it can be soluble in organic solvents. Okay, like alcohol. Okay, example, natural oils, waxes, and steroids. Okay, these are the examples of lipids and fats, oils, and these are not soluble in water. Fatty acids now. A fatty acid is a carboxylic acid with a long aliphatic tail. Aliphatic means along. Okay, well, aliphatic means aromatic means with the help of the benzene ring. Aliphatic means a linear or branched, but there will be no aromatic ring. So aliphatic tail chain, which is either saturated or unsaturated. Okay, so saturated means where there will be no double bonding, and unsaturated means where there will be double bonds, like it has been shown with the red circle here. This is the unsaturated, and here it is the saturated. Okay, so fatty acids is the carboxylic acid with long aliphatic chain or tail, which is either saturated or fatty. So a fatty acid has a carboxylic group attached to an R group, saturated fatty acid. Okay, saturated fatty acids have no double bonds between the individual carbon atoms of fatty acid chain, like I told you, and unsaturated fatty acid have one or more double bonds between the carbon atoms of fatty acids. Okay, there can be triple bond also. Okay, so double bonds or triple bonds between the two or more, or one or more. Okay, so that is the unsaturated fatty acid. The R group be a methyl or ethyl or higher number of groups, 1 carbon to 19 carbon. So it could be methyl, ethyl, or it could be any members or higher groups of CH2 up to carbon. Okay, 1 carbon to 19 carbons. Okay, for example, palmitic acid has 16 carbon including carboxyl carbon. So 16 carbon, palmitic acid. Okay, so this is the palmitic acid which has got 16 carbon. You can see. Okay, fatty acids with 16 carbon. Okay, arachidonic acid has 20 carbon atoms having 4 double bonds including the carboxyl carbon. You can see the 4 double bonds. Okay, and 20 carbons. So it is the arachidonic acid. Arachidonic acid, another fatty acid. Now glycerol is another compound. Simple lipid is glycerol, which is trihydroxypropane. Trihydroxypropane. Okay, the technical name, the technical name of the glycerol is the trihydroxypropane. Okay, it is a simple lipid. the simplest you can see. You can see the trihydroxy. Okay, so 3 carbon atom, so trihydroxy, you can see the HCOH, right? So here trihydroxy propane, because 3 carbon are there, so it's propane. Now lipids classified into lipid as the, you can see, as the simple lipids, compound lipids, and derived lipids. Okay. Now you can see in the simple lipids, it is the esters of fatty acids. And these esters of fatty acids are again divided into fats and oils. Fats are the simple lipids. Okay, when this means they remain solid at room temperature, and oils are at liquid state at room temperature. That is the difference. So simple lipids are esters of fatty acids, and these are classified as fats and oils. Okay, fats remains solid at room temperature, and oils are in liquid condition at room temperature. Okay, compound lipids, these are the esters of fatty acids and alcohol containing either groups also, other groups also. So that means ester of fatty acids, okay, and alcohol containing other groups also. So simple lipids included the esters of fatty acids, but in case of the compound lipids, esters of fatty acids and alcohol contain other groups also. Okay, so this is the, now in case of derived lipid, composed hydrocarbon rings and a long hydrocarbon side chain. Okay, we find this hydrocarbon ring, okay, and a long hydrocarbon side chain is also found. That is the derived lipid. You will see the simple lipid, ester of fatty acids, and a long chain of alcohol. Simple lipids, fatty acids, glycerol, triglycerol. Try to understand this. Try to understand this. Esters of fatty acids, okay, and long chain alcohols, that form the glycerol. Fatty acids, okay, here the fatty acids, and the glycerol, now forming the triacyl glycerol, forming as triacyl glycerol. Example is the waxes, fats, oils. Okay, so these are the examples of the simple lipids. Okay, triacyl glycerol. Okay, waxes. A wax is simple lipid which is an ester of a long chain alcohol and fatty acid. So long, like we observed. Okay, so and in case of the oil, triglycerides that are liquid at room temperature are called oils. These are the triglycerides that remain liquid at the room temperature are oils. Fats, triglyceride that are solid at room temperature are called fats, which I told you earlier. Now monoglyceride. Monoglyceride is a glyceride in which each glycerol molecule has formed an ester bond to exactly one fatty acid molecule, glycerol plus one fatty acid. Okay, so here monoglyceride, fatty acids plus glycerol, you can understand. So it is the monoglyceride. Okay, right, monoglyceride, glycerol plus one fatty acids. So one fatty acid here and the glycerol. You can see this part is the fatty acid and here is the glycerol, and this is the glycerol. Okay, so glycerol plus fatty acid is monoglyceride. Okay, diglyceride, again there will be two fatty acid, one fatty acid and two fatty. So these are the two fatty acids and one is the glycerol. Okay, so that is forming as the diglyceride. Okay, a diglyceride or a diacyl glycerol is a glyceride consisting of two fatty acids chains covalently bonded to a glycerol molecule through ester linkage. This is important, through ester linkage. They are bonded through the ester linkage. Okay, and this is called diacyl glycerol or diglyceride. Now triglyceride, this is the important, found in the animals and the living organisms. A triglyceride is an ester derived from glycerol and three fatty acids, and we can see this is the triglyceride, and its biological importance is there. R1, R2 and R3 are these fatty acids, okay, and there is the glycerol. Here is the glycerol. Okay, it forms the triglyceride. Phospholipids. A phospholipid is composed of two fatty acids, a glycerol unit, a phosphate group, and a polar molecule. You see this. See here, a phospholipid. So the main the reason, or you can see the property that is deciding here is also composed of two fatty acids, a glycerin and a phosphate group. So this phosphate group is making little different, okay, and a polar molecule is there. So this is the phospholipid, okay. You can, yeah. So this is phosphorus is there here, and this makes the it's little different, okay. This presence of this phosphorus, okay. So this is called phospholipid. Two fatty acids are there, okay, and a glycerol unit is there, and then a phosphate polar molecule, okay. So this is cephalin. This example is a cephalin. Phospholipid is a group name, and this example of phospholipid is the cephalin, okay. Okay, lecithin. This is the lecithin. So these are the, this comes on the complex little because they have the little they are the derivatives, okay, and they have got complex structure. They are found in cell membrane, okay. Our cell membrane or plasma membrane is made up of lipid bilayers, and this lipid bilayer is made of the phospholipid, so found in the cell membrane, and plays a very important role, okay, in the exit and entry of the substances, and also other functions. Neural tissues have lipids with more complex structure. The nerve tissue, neural tissue, neurons have more lipids with more complex structure. Okay, so heterocyclic compound are the compounds are cyclic organic compound. Typical heteroatoms include nitrogen, oxygen, sulfur. Okay, living organisms have a number of compounds in which heterocyclic rings can be found. Heterocyclic, not so biological compounds having heterocyclic rings in their structure. That is the purine, adenine, okay, structure, or that is found nitrogenous base found in DNA and RNA. So they are the heterocyclic rings in their structure. Purine, adenine, guanine, pyrimidine, cytosine, thymine, uracil. These are the bases, nitrogenous bases in DNA and RNA, and they have got heterocyclic rings in their structure. Okay. So what are nucleotides? Nucleotides are molecules comprised of a sugar molecule, a phosphate unit, and a nitrogenous containing base. We'll see this one. You see, adenosine, guanosine, thymidine, uridine, and cytidine are the nucleosides. Okay. Here, nucleoside means nitrogenous base plus sugar. Because in case of nucleotide, nucleoside plus phosphate. So forming a nucleotide, we have got nitrogenous base, sugar, and a phosphate. But in case of nucleoside, it is nitrogenous base and sugar only, no phosphate. Okay. So here you can see there is a sugar, okay, there is a phosphate and nitrogenous base. So this is a nitrogenous base, here is the sugar, this is the sugar, and this is the phosphate group. So this is forming the nucleotide, and here it is in the, so when we are exiting, we are not including this part, okay, up to here, okay. So this part, if you say, this becomes the nucleoside, but if you include this also, then it becomes the nucleotide whole. Okay, so that means we are excluding, if we are excluding the phosphate, then it is a nucleoside. When including the phosphate along with the nitrogenous base and sugar, then it becomes the nucleotide. Okay, nitrogenous base. Adenylic acid, adenosine monophosphate, AMP, also known as 5-adenylic acid, is a nucleotide that is used as a monomer in DNA and RNA. This adenylic acid, okay. So adenine and guanylic acid, guanosine monophosphate, GMP, is also known as 5-guanylic acid, is nucleotide that is used as a monomer in DNA and RNA. Okay, and this is look like this here. Okay, it's a monomer, okay, in DNA and RNA. It's used. Now, adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acid are nucleotides. Nucleic acids like DNA, RNA constitute nucleotides only. Okay. DNA and RNA functions as genetic material, that you already know that, because the part of the different certain part of the DNA that acts as the a gene, and they are controlling they are expressing some characters, and they produce responsible for production of some certain protein also. So lipids are insoluble in water, organic solvent, water and organic solvent. So it is the organic, it is water, number one, water. Soluble in, sorry, insoluble in water, but soluble in organic solvent. Soluble in organic, soluble. So in water they are insoluble, but in organic solvents like in alcohol they are soluble. That is dissolved. Okay. Triglycerides that are liquid at room temperature are fat. Is it? That is the fat. Liquid, sorry. The liquid at room temperature is oils, and solid at the room temperature is the fats. Okay. Fats, oil, waxes, and steroids. Triglycerides that are liquid at room temperature is oils, and fats is the solid at room temperature. Glycerol is 1,1,1 trihydroxypropane, or 1,2,2,2 trihydroxypropane, or 1,2,3 trihydroxypropane. It is 1,2,3 tri, because there are three positions: 1 carbon, 2 carbon, and 3 carbon. So all 3 carbon. First 1 carbon, then 2 carbon, and 3 carbon. So it will be 1,2,3 trihydroxypropane. Okay, this is the correct answer. Number of carbon atoms in the R group are arachidonic acid. It is the 19, or 20, or 16, or 15. It is 20. Arachidonic acid is 19. Okay, it is 19. 19 carbon atoms are there in R group of arachidonic acids. So we find up to 1 to 19 or 20. So this arachidonic acids, or the oleic acids, okay, stearic acids, and some different types of fatty acids are found. Okay, some are saturated, some are unsaturated, and these are present in the different different lipids and fatty acids in organisms, in animals' body, okay, and in plants also. Okay, oleic acid, arachidonic acid. Our nucleoside is made up of sugar and phosphate. No. Sugar and nitrogenous base, phosphate and nitrogenous base, or sugar, phosphate. No. The fourth one, sugar, phosphate, and nitrogenous base, is the nucleotide. Phosphate and nitrogen is the correct answer. That is number 3 is the correct answer. Okay. Sugar and phosphate. There is no phosphate. Sugar and no, not number 3 is also not correct answer. Phosphate is not there. Nitrogenous base and sugar is there in nucleoside. So 3 is not correct answer. This is also not correct. This is also not correct. This is also not correct, because phosphate. So this is the correct one. Okay, we'll see. Yes, sugar and nitrogenous base is the correct answer. In case of nucleotide, then it will be sugar, phosphate, and nitrogenous base. Okay. Which of the following can function as genetic material? DNA, or RNA, or both 1 and 2, or lipids? It is both 1 and 2. Normally, DNA is the genetic material, okay, found in many organisms, but in very few, like virus, okay, we find that RNA also acts as the genetic material. Okay, so both RNA and DNA acts as the genetic material. Okay, so this was all about the amino acids and the lipids, and we started with this topic, amino acids, that we studied about the amino acids are made up of the it is a modified form, you can say the substituted form of the methane, CH4, where we found that there is one amino group replacing the hydrogen, there is one carboxylic or acidic group replacing the hydrogen, and there is hydrogen, and there is the replacement of hydrogen with the R variable R group, alkyl group. Okay, and thus the amino acid monomeric unit is the structure for the monomeric structure for the formation of protein. When proteins are formed, these amino acids are joined by peptide bond. I told you many times. Now these amino acids are of different types. Okay, and there are 21 amino acids that constitute that forms the protein, and in organisms, in living organisms, the different proteins are made up of the different permutation and combination of these 21 different amino acids. Okay. Then we studied about the lipid. Okay, lipids are, you saw that there are different types: simple, okay, compound and derived, okay. And in case of simple, there is the esters of fatty acids, and they are then again these are different. Simple are divided into fats and oils, okay. And there is a glycerol and monoacylglyceride and diacylglyceride, diglyceride and triglyceride. Triglyceride being the most important because these are found in the living organisms, okay. And then we study about the nucleotide and nucleoside, okay. When there is a nitrogenous base, and then there is the sugar, okay, then it's called nucleoside. And when there is a nitrogenous base, phosphate group and sugar group, all three, okay, then it is called the nucleotide. DNA and RNA are the genetic material. DNA is the mainly genetic material, and these nitrogenous bases are found in DNA and RNA, okay. So this was all about the biomolecules like amino acid, nucleoside, and lipids, okay. In the next class, we'll study in detail about some other biomolecules, okay. And this is one of the important topic. If you study nicely, this was the all important concept, okay. And in the next class, we'll study the another pattern in more detail about some important discussing along with the some important questions also, okay. So tomorrow again we will meet at the same time with another topic, another biomolecular topic, and we'll study in for that. And so we'll meet again tomorrow at the same time, okay. So thank you for joining and listening today. Tomorrow we'll meet, and so till then, goodbye and take care.